Yeah.
Perfect. Okay. All right. So thank you again, everyone, for having me and for attending this, I would say, unique webinar. I haven't been asked to do one before on trials specifically. All the other webinars I have done have been about the diseases in general, right? MPNs, various aspects. But I think this is very unique because as patients and caregivers, you need to be aware of what trials are out there, and so that maybe you can seek out centers that have those trials, and so very happy to do this today. So the way I have this formatted is that I only have slides on trial designs, and they certainly tend to be quite dense. As you can see here, they can be pretty detailed. But what I'm going to try and do is walk through the three conditions, ET, PV, MF, in the context of trials, and sort of give you the big picture on why the drug is exciting, why do this, why participate in this, and then perhaps spend less time on the details of the design, which certainly, if you do pursue one of these, you'll certainly learn all those details. So with that sort of general direction, let's get started with ET, and then we'll go into PV for a little bit, and then finish with MF, where most of the action always is. Just because MF is a worse disease than ET and PV, and so there's generally more trials, more drug development in the MF space. Although I think what's happening now is that people are realizing that if we can really come up with disease modifying treatments in the ET and PV phases, we can probably maybe even prevent MF, right? So the whole the I think the paradigm is shifting. In ET and PV, we used to think, let's just try and reduce the risk of blood clots, but now it's beyond that. There's a lot of efforts at disease modification, prevention of progression to MF, to AML, that's going on in ET and PV. Dalton, let me just confirm. Are you seeing the first slide, the short span 006?
Yep, I am.
Okay, perfect. So short span 006 and then 007 on the next slide are the Merck-sponsored phase 3 trials of bomedemstat. Bomedemstat is an LSD1 inhibitor, lysine-specific demethylase 1, LSD1. Now, this is an epigenetic target. So, again, at a very superficial level, or in a very, perhaps in a very broad sense, the we have a number of epigenetic modifiers, we call them. There's a number of genes which may be mutated or not in these diseases, but they all have epigenetic functions, and epigenetic functions are functions where these genes, so LSD1, for example, EZH2, ASXL1, TET2, you may have seen some of these on your molecular panel when your doctor gets an NGS, next generation sequencing panel, on your bone marrow or blood. You may have seen beyond the three driver mutations, JAK2, MIPL, and CALR. You may have seen some of these genes being mutated. Now, while they can be mutated, they can also have important functions if they are not mutated. So LSD1, for example, is not usually mutated. I've never actually encountered a mutation in LSD1, but nevertheless, it's a very important epigenetic modifier, as they are called. So these genes control the expression of other genes. That's really what epigenetics means. They control what genes are expressed, what genes are silenced. So LSD1 is one of them. And bomedemstat is a small molecule inhibitor of LSD1. And there's been phase 2 trials. This drug used to belong to a company called Imago that was acquired by Merck. And there's been phase 2 trials showing some really nice results in ET. So control of the platelet count, control of the white count, stabilization of hemoglobin, so not dropping the hemoglobin, which you would not want, improvement in symptoms. So all of that has led Merck now to embark on 2 phase 3 trials. So you're looking at one of them on this slide, ShoreSpan 006, This is in patients who have had an inadequate response to or not been tolerant of hydroxyurea, which remains the most commonly used frontline drug in ET in the US. And so patients who are sort of failing that can go on this trial. What I like about this trial is that look at the comparator arm in the green box towards the middle. So it's bomedemstat versus best available therapy, and that's a really broad array of best available therapies to choose from. So enegrelide is on there, busulfan is on there, not that that's used very much, but it's on there. Interferons are on there, ruxolitinib is on there. I was very impressed to see that, because these are all really helpful drugs that we all use. So I think if I were a patient going on this trial, I wouldn't worry too much about ending up on the best available therapy arm, because that could happen, right? These are controlled, randomized trials. So certainly that could happen, but you're still getting a very decent, a very acceptable therapy. So I'll move on to the next slide. Oh, okay. All right. Is it moving, Dalton? 007 now?
Yep, I see 007.
Okay, perfect. Thanks. So this is the kind of a sort of a mirror image, but this is in the front line. So it's the same drug, bomedemstat, the LSD1 inhibitor, but here it is compared to hydroxyurea. So in a newly diagnosed patient with ET, or I should actually, to be correct, I should say cytoreductive therapy naive. So somebody who has never had cytoreductive therapy for ET can go on this. It's bomedemstat, the LSD1 inhibitor, versus hydroxyurea. Again, hydroxyurea being the most common frontline cytoreductive drug in the US was chosen as a reasonable comparator. Now, granted, a lot of people may increasingly may be using interferon in frontline, but interferon is not specifically approved in ET the way it is in PV, for example. So hydroxyurea, I think, is still by far the most commonly used option. And so this is a comparison of bomedemstat against hydroxyurea. All right, so those are the short span trials. Phase 3, remember, bomedemstat, an oral drug, epigenetic modifier, LSD1 inhibitor versus either best available therapy in second line or hydroxyurea in front line. All right, so there's a lot of excitement now about mutant CALR, targeting mutant CALR, and these are, CALR mutations, as I'm sure our audience knows, is the second most common mutation in MPNs after JAK2, and CALR is present in mutated in, I mean, in 20 to 30% of patients with ET and also MF. It's usually not seen in PV. So Insight, as is shown here, they are developing or are studying, I should say, a mutant CALR antibody. It's IV, it's given every 2 weeks. So what happens with CALR, and this is just a principle that perhaps may help explain why this is a unique target, is that the mutation causes the CALR protein to come outside the cell, okay? So CALR normally stays inside the cell and does what it needs to do, but the mutation causes it to come out. And then it binds to a receptor called MPL, which is the thrombopoietin receptor, so also called TPOR. And then it does it does what it does to cause the disease, right? To drive the ET or MF or whatever it might be. But that process of CALR coming outside the cell makes CALR uniquely susceptible or vulnerable to immunologic targeting. So if it stayed inside the cell, antibodies would not be able to see it. But because the mutation brings it outside the cell, antibodies or immunotherapy in general can actually see it and target it. So this is an antibody that does exactly that. So busy slide. Again, like I said, I won't go too much into the details of inclusion and exclusion criteria for any of the studies, but this is the study design. It enrolls ET as well as MF. So I'm just showing this together while we are talking about ET, but remember it also includes MF, as you can see. There's treatment group A, that's MF, treatment group A for ET as well, and then there's a treatment group B for people who've been on Jakafi or ruxolitinib and are having a suboptimal response. They can add on the antibody. That's not open yet in the US, by the way, but the treatment group A, both of them, the MF and the ET, definitely are open. And then if you look to the right of the slide, that I believe has just opened this month. That's this part 1C, where patients who have not had any JAK inhibitor, essentially have not been treated for their MF, can also get on this. This is new. I'm told this is as of this month, and they can start the antibody. And as you can see, again, if you focus on the right of the slide, if the antibody itself is not leading to an optimal response after 12 weeks, they can add a ruxolitinib or Jakafi. So this, again, complicated study, but very exciting molecule. The first results in ET were presented this past June at the EHA Congress in Milan, and has certainly added to the excitement around this. So key eligibility criteria, again, I'm just staying on the ET for this portion of the discussion. And so if you'll focus to the upper left, of course, you have to have a CALR mutation. Remember, this is not for any other patients. This is for those whose driver mutation is the CALR. Like I told you, ET or MF. But for the ET part, anything that qualifies as high risk. So being over 60 years of age, having had a prior clot, having had significant bleeding from the ET, and also having had a prior therapy. So just to be clear, right now, because it's early, this trial is not yet for newly diagnosed ET or untreated ET. It's not for them. It's for previously treated ET, at least one prior treatment. But for the MF, as I said earlier, and I know this can get a little confusing because the same trial has so many arms, but for MF, starting this month, there is this ability to enroll patients who have not had a prior JAK inhibitor, who can just start on this and then have a JAK inhibitor added if needed. All right, so now moving to a different drug, also from Insight, but this is what is called a bispecific antibody. So again, both of them are antibodies. Both would be IV, but the first one is a straight antibody, and it doesn't necessarily use the help of any other entity. But the bispecific antibody, the idea here is that the antibody binds to CALR, the mutant CALR. And remember, I explained that the mutation brings the mutated CALR protein outside the cell so that the antibody can recognize it, right? So the antibody binds to that, but also at the same time, simultaneously, it binds to T cells. T cells, which are part of our immune system, lymphocytes, T lymphocytes, and it uses the ability of the T cells to kill target cells. It uses that ability to kill the CALR mutated cells. So it's latching on to the CALR mutated cells, but not killing them on its own. It's using the T cells to kill them. Now, this, I think, is literally just opening. There's no results. There's no real experience with this. I think it's just literally opening, but just something to be aware about. And also, I don't have slides on this, but Janssen has a similar molecule, also a bispecific. These are called T cell redirecting antibodies or bispecific antibodies. Janssen has one that's been ongoing for a number of months now. I just don't have slides on that one, but conceptually they are similar. And so I'm showing you this one, which is just opening, like I said. And that's just more on this particular study design. So again, these will be for ET and MF. Remember, CALR is not really a player in PV. All right, so moving on to something different, a little bit different, but same sort of theme, We were talking about how to target mutant CALR. Now we're going to talk about how to target mutant JAK2. So we have 4 JAK inhibitors on the market, but remember, none of them is selective for the mutant JAK2. They're all going to hit mutant as well as the normal JAK2, what we call wild type. Um, Yet, JAK2 is the most common driver mutation. It's about 60% of ET and MF, 95% of PV. So it's by far the most common mutation. So there's a lot of interest now in selectively targeting the mutant JAK2 and sparing the normal JAK2. And that's what's shown here. This is also Insight. It's also an Insight drug. So what they're trying to show you in these cartoons is that the JAK2 protein has a number of domains. So one of them is the kinase, which actually is the engine, is what does the work of JAK2. But there's also the pseudokinase, which keeps the kinase in check. So when you have a mutation, this V617F, that is by far the most common mutation in these diseases, that pseudokinase is no longer able to keep the kinase in check. So the kinase is just going, and that's what drives the disease. So this drug, this Insight drug, and perhaps other companies, many others are interested in this. Perhaps there'll be others that will enter the clinic. These drugs bind to the pseudokinase, not to the kinase, to the pseudokinase, where the mutation is. And the expectation is, I'm sorry, let me turn this off. The expectation is that they will just block the mutant protein and not the normal protein. So again, this study, lots of details on this, but remember, this study is actually open for ET, MF, and PV, because JAK2 is obviously a mutation that is common to all of them, especially PV. So, but they all require one prior therapy. So this drug is not yet at a phase where they are confident of giving it to non-JAK inhibitor treated patients or previously untreated patients. This is still for patients who've had at least one prior therapy. All right, so moving on, as you can see, we talked about ET. We sort of also got into PV kind of subtly with some of those overlap trials there. So now we'll get into MF. Now, and another thing just to note, maybe I should have said this earlier, there are a number of phase 3 and other major trials that just finished accruing, or that just had results come out. Some examples are, Surpass ET, the phase 3 trial of ropeg interferon alpha 2b in ET, or the selinexor and ruxolitinib trial in MF, or the imetelstat trial in MF. Now, I'm not mentioning those or showing slides on those because those are done. And like I said, either results awaited or results just released. So I'm not showing those. So coming back to this slide. So in MF, this is a multifaceted disease. Some patients, spleen is their problem. Others, symptoms are their problem. Others, anemia is their problem. Some, unfortunately, all three are the problem, or two of the three. So it's a very heterogeneous disease. So there are, I'll mention in 2 slides, some studies that are ongoing for anemia, just for the anemia part of it, okay? So this is DISC. Oh, I'm so sorry. Forgive me. We're still on PV. There's one study here on PV. Let me do this and then get into MF. Sorry about that. Premature jumping into MF. Okay, so this is PV. So scratch that for a moment. This is PV. So this is a drug called DISC 3405. Okay. So let's talk about this concept for a little bit because I've not touched on this at all. So recently, you may have heard there was a lot of news around a drug called rusfertide. This is a hepcidin mimetic. Very positive phase 3. Yet another example of what I was just saying, where we had some major phase 3 trials read out this summer or just this past summer, or just finished accruing last month. Rusfertide was one of them. And this is a drug that's highly likely to be approved, we think, because of how positive the trial was. And this is a hepcidin mimetic. So a hepcidin mimetic, the idea is that in PV, okay, we're on PV. So in PV, if you mimic the action of hepcidin, or you increase your own hepcidin, what happens is that the iron gets sequestered or trapped in the body, in the spleen and liver, and the bone marrow is sort of starved or deprived of iron. That's the principle, that if you mimic hepcidin or induce your own hepcidin, then you're depriving the bone marrow of iron because you're trapping it and storing it away. So that was the excitement about rusfertide, and more to come on that. So now if you'll focus on this slide, this is DISC 3405. It's a hepcidin inducer. It's an antibody against something called TMPRSS6. But again, what's important to focus on, really the big picture, is that it will increase your own hepcidin. So the idea here is, let's increase hepcidin, or mimic its action with rusfertide, and thereby deprive the bone marrow of iron. And so we can make less red blood cells, and so less phlebotomy, maintain that hematocrit under 45, like you know it needs to be kept. That's the idea. Okay. So there are several like this, but this is the ongoing one. What I tried to do today is only have slides on things that are currently accruing, and not on things that are coming or that just closed. Okay, so now MF. All right, so this is a drug that's actually familiar to some of you, I'm sure, who have PV, or who have family members with PV. So Ropeg interferon alpha 2B, or Besremi, is an approved drug for PV. And I just alluded to it being in a positive phase 3 trial for ET. We just learned this past June that the phase 3 trial in ET was positive. So this is the same drug, Ropeg interferon alpha 2B, trade name Besremi. So this is now entering a trial in MF, in early MF, and that's what I'm showing you. Because early MF, patients who have good blood counts, spleen not very large, no symptoms or mild symptoms, those patients are often observed. Those patients are not always given treatment. And so this is really a much, I think an unmet need that this trial will fulfill, that, the goal being, can we intervene early in these patients who are otherwise being observed and not being given a JAK inhibitor because their spleen is not that big, they don't have symptoms, counts are pretty good. And can we go in with interferon early and see if we can change the natural history of the disease? And this is just more on that. Okay, you'll basically see these are people with good blood counts. They don't have severe anemia. They don't have a very big spleen, things like that. And people who would be reasonable to observe because the comparator is placebo. So it's not an active comparator, it's placebo, because in many of these patients, they are not given anything. So whether that's right or not is another debate. But the point is that many of these early MFs are often watched, and observation is actually an acceptable option, even in the in international consensus guidelines, and that is where this trial is aiming to make an impact. All right, so now I'm getting into the more, shall we call it, the more the MF that we are more accustomed to thinking of, big spleen, symptoms, blood count abnormalities, maybe some blasts, the overt MF, and the MF that clearly needs treatment. So there is this concept of blocking a protein called MDM2. And MDM2 is a protein that we all have in all our cells that basically keeps the function of P53 in check. And P53 is a gene that kills cells. It causes apoptosis, or it basically pushes cells over the edge to their death. And in cancer cells, P53 often doesn't work. And that's why cells don't die. So, and that is due to mutations or deletions of P53. Now, fortunately, in MF, that is rare. P53 usually works. It's not mutated or deleted most of the time. And so the idea here is that if P53 is basically okay, what can we do to enhance its function? Okay, so if you block MDM2, you're going to make P53 more active. You're going to unleash it. And that is the idea here of blocking MDM2 and making P53 more active. Now, just some words on this trial. This is a unique trial in the field. There is no other trial like this ever or currently that is quite designed this way. So this is a trial called POIESIS, that the name is on the upper right, where you sign consent before you start Jakafi or ruxolitinib or any JAK inhibitor. So patients who've not started on a JAK inhibitor, they sign consent, they enter the trial, they start ruxolitinib or Jakafi, and after 18 to 24 weeks, their response is evaluated. And if they have a suboptimal response in terms of their spleen reduction, their symptom reduction, only if they have a suboptimal response at that point, that's when they get to add naftamadlin. And I was just describing what this is. It's an MDM2 blocker or inhibitor or placebo. Okay, so everybody starts on Rux. After 18 to 24 weeks, if they are suboptimal, they add naftamadlin or they add placebo. All right, so selinexor is another exciting drug. This is a drug that actually, just completed in August their large phase 3 trial in combination with ruxolitinib or ruxolitinib and placebo. So that was a big major frontline trial, ruxolitinib and selinexor versus ruxolitinib and placebo. That's done. This is the Sentry 2, sort of the sequel. This one is unique also in the sense that it does not start with a JAK inhibitor. It starts with selinexor, which we know in other trials, in like earlier phase 2 trials, is a very active drug, especially for spleen, actually also symptoms, but more spleen. And so we know it works on its own. But what is different about this is that typically in this field, we are all used to starting with a JAK inhibitor. Here it's different. You start with selinexor, and after 12 weeks or after 24 weeks, if you don't meet certain benchmarks, you add the JAK inhibitor. So there's no placebo here, okay? There's no placebo here. That contrasts it from the previous trial. But in some ways, they are kind of similar in the sense that you start with one drug and add the other one later. With the previous one, remember, POIESIS, you start with ruxolitinib, the sort of the standard medication, and you add naftamadlin or placebo. Whereas with this one, you start with selinexor, so a little bit unconventional in that sense. And then later you add the JAK inhibitor if you're not where you want to be. Just inclusion and exclusion criteria. This was originally designed to target patients with a lower platelet count, people who may not be ideal for high doses of ruxolitinib because of their platelets. And one nice thing about selinexor is that it doesn't really suppress the blood counts. So the idea was that, with these patients with somewhat lower platelets, could we, look at this, starting with this and then adding a JAK inhibitor as necessary? But again, trials are always amended, and that platelet count, criterion could change. But this is how it was originally designed. Now, okay, so this is what I thought I was jumping to some time ago. All right, so this is anemia. So remember, I started to talk about anemia, and I was saying that there are patients with MF who just have anemia, or who may have a little bit of a enlarged spleen slash symptoms, but whose main issue is anemia. They are tired all the time. They are needing transfusions. Hemoglobin is low, and that's kind of their main issue. So there are two trials for people like them, to my knowledge, that are currently open. So this is Odyssey. Odyssey is two known drugs coming together. So momelotinib, that's, as you know, is a GSK drug, approved for the last 2 years now, and luspatercept, a BMS drug, not approved for myelofibrosis, but approved for MDS, widely used. We've all used it in myelofibrosis too because it works, and we have good phase 2 data showing that it works. So this study brings them together. So momelotinib plus luspatercept in patients who are transfusion dependent. So they have to be transfusion dependent. That part is required in this particular study, or a hemoglobin less than 8, because less than 8 is like being transfusion dependent. That counts as well. And they can have had a prior JAK inhibitor or not. They could also be JAK inhibitor naive. So both sorts of patients are welcome, and this is transfusion dependent patients, like we just said, or hemoglobin less than 8. Momelotinib plus luspatercept, the two companies have nicely come together to provide the drugs, which is obviously important. This is the other anemia study I'm aware of. And again, remember, there are other drugs, on the horizon, which are not open yet in the US, and I'm not getting into those. But this is a drug called DISC-0974. So we talked about a DISC drug, if you remember, a few slides ago, that was for PV. That was where I said we are trying to either mimic or induce hepcidin and reduce iron for the bone marrow and help our patients with PV. This is the exact opposite. The exact opposite. Here, we're trying to help our anemic patients with MF. And what we are doing here is we are actually blocking hepcidin. This is a mechanism it's an antibody against something called hemojuvelin. And ultimately, the idea is to reduce hepcidin. So less hepcidin, so the opposite of what you want to do in PV, less hepcidin will mean that the iron is less trapped or sequestered and more available to the bone marrow. So again, the exact opposite of PV. So if it's more available to the bone marrow can use it and make more hemoglobin, make more red cells. So that is the idea with this one. Okay. These are all, this study, we've had results presented a few times. It's a very active drug. So certainly optimistic about both this one and the previous one, which, remember, I told you these two drugs they don't need any introduction. They are already active drugs. They're just coming together. And then this is a new drug, but an active one. And so excited to see what this trial ends up showing. All right, so done with anemia. We're very much into MF right now. A couple more concepts, but this is no longer about anemia. So now it's more about how can we address the disease holistically? So we like all aspects of the disease. So we already talked about naftamadlin. We talked about selinexor. Those things can do that. So here's another drug that appears to, at least in animal models, have effects broadly on the disease. And this is called nuvisertib. Nuvisertib, you'll see it on the next slide, belongs to a company called Sumitomo, and it blocks a target called PIM, as you see here, PIM1 to be exact. And in animal models, it was very effective in making the fibrosis get better, shrink the spleen, bring the blood counts down, etc. So this is the study. It's ongoing. We've already seen results on 77 patients. And again, this is continuing. It's a phase 1/phase 2. And important to mention, they now have confidence about the drug's safety and efficacy to the point that they are now starting to combine. So this drug is being combined with momelotinib and ruxolitinib. So to my knowledge, probably right now, other than luspatercept, which I just showed you, the only other drug to be combined with momelotinib in a clinical trial. Of course, there are many that have been combined with ruxolitinib. So momelotinib, again, for our viewers, Ojara, the most recently approved JAK inhibitor. So novicertib is being combined with both Ojara and Jakafi. All right, finally, I'll probably end with this concept, and looks like we're doing okay on time. So this is a very neat concept, and I'll show you where this is leading to. So we have 4 JAK inhibitors marketed, right? We have ruxolitinib or Jakafi, fedratinib or Inrebic, pacritinib or Vonjo, and momelotinib or Ojara. They're all type 1 JAK inhibitors, which bind in a certain three-dimensional configuration or conformation. But there's also a thing called type 2 JAK inhibitor, and these bind in a different configuration. So back in 2012, the laboratory of Ross Levine at Sloan Kettering in New York had shown that one of the ways that cells become resistant to the drugs we have today, the type 1 JAK inhibitors, is something called persistence. Now, that's a complicated phenomenon. I won't go into details of that. But basically, what it is that the drug is bound to its target, right? The drug is there. It's not like the drug's not there. You're giving the drug, the patient is on it, it's bound to its target, but the cells find a way around it. So the JAK family has multiple members. So if JAK2 is blocked, they just use other members to keep signaling. And this phenomenon is called persistence. And they were able to show, this is back in 2012, that if you use a type 2 JAK inhibitor, then you're able to overcome that phenomenon, the persistence phenomenon. And so they looked at certain, in the lab, they call these tool compounds, the drugs that they use in the lab to prove a principle, but that doesn't necessarily mean that it can be used in people. Like it could be toxic or whatever. And so they use this drug CHZ868, but that was never going to be a human drug, a drug for human beings because of toxicity. So now there is such a drug, and it's called AJX. I'm sorry, this is just the name of the trial. The drug is called AJI 11095. So this is from a company called Ajax, hence the AJX, and this is a type 2 JAK2 inhibitor. Okay, so again, all the 4 we have today are type 1. And the idea here is, okay, that's not working anymore. Let's try a type 2, which binds in a different way and should be able to overcome that persistence phenomenon that I just explained to you. So this is a phase 1 trial. It's ongoing, and early days, no results yet, but just a very, very unique and very scientifically, intellectually exciting concept. And I will end with that. Happy to take questions. Thank you.
Thank you so much for sharing all of those. And we do have a couple of questions, so we're going to go ahead and jump into that. But it's so exciting to see all the work that's being done, and just really how far the field has come, and how much further it has to go is incredible. So our first question that we have is coming from Beverly, and they're asking, is there any research or studies for ET with both the JAK2 and a CALR mutation present?
So what happens is, it's a great question, because this is a rare situation, right? But it can happen. We've all, I think, seen these cases where there's 2 driver mutations. And what usually happens, though, and I urge you to go back and look at your report if this is your situation, usually one will be predominant. Of the 2, the VAF, or allele burden or variant allele frequency, of one of the 2 will be very high and the other will be very low. So in principle, I don't think that you'd be excluded from the mutant CALR or the mutant JAK2 just because you have both. as long as one of them is the main driver. And I would urge you to look at the studies because I want to say we've encountered such a patient as well recently, and I do not believe that that would exclude you either mutant JAK2 trials or mutant CALR trials.
Okay, that's good to know. Then next up, we have Selena asking, when you define the spleen as not too big for certain trial drugs, can you please be more specific about the degree of enlargement? Is there a certain size limit or parameter that you're looking at for that?
Yeah, that's a fantastic question also. I think the context in which I said this was the Pharma Essentia Hope PMF trial, the phase 3 trial where they are looking at ropeg interferon alpha 2b or placebo in
Mhm.
early myelofibrosis, right? And I may have made the comment is that this is for patients where the spleen is not very enlarged, etc. So in that trial, and it was buried in the busy slides I showed you, in that trial, the spleen has to be smaller than 5 centimeter by hand, by palpation. So that is as far as that trial. But I think this question raises a much bigger debate, which is should we be just watching patients who have some degree of enlarged spleen, but not yet 5 centimeter? Okay. So, 2 centimeter, 3 centimeter. I think no. I think we've learned enough to feel that we should probably be intervening in those patients. However, the trial is justified because if you look at guidelines, they still do recognize observation as a legitimate option for patients without symptoms with a small or undetectable spleen. And because observation is still practiced and still is recognized as okay, the study is has decided to compare the drug ropeg against placebo. Okay.
Mhm.
Now, whether each individual patient, each individual doctor uh, looks at it that way is a different question, right? Because you could argue that with any degree of spleen enlargement, you would want to start ruxolitinib, but that is a different question.
Okay. And then next up, we have Sylvia asking, are JAK2 type 2 inhibitor trials only for myelofibrosis?
Yes. So right now, the Ajax trial, which I showed you at the end, is only for myelofibrosis. It is not for PV or ET yet. That's correct.
Okay. And then we have another with the Ajax, so I'm just going to roll into that one. But do you know kind of what the early results of those Ajax studies are? Or is it too early to really report on yet?
Yeah, there are no results in the public domain on the Ajax study. So yeah, really nothing that I can comment on. It's in dose escalation. So you know how they go through
Yeah.
different doses and determine safety and early signals of efficacy, but that's ongoing. There's no results yet.
Okay. Good to know. And then next up, we have an individual asking, how would you approach a patient with early myelofibrosis, limited fibrosis, no enlarged spleen, blood count relatively normal and under control, but with a high JAK allele burden and a TP53 mutation, detected almost 2 years back and hovering around a 10% VAF. So early myelofibrosis, but do you think clinical trial might be a good option for someone like this, or is observation probably a good step?
So if the spleen is not enlarged at all, right? We're not saying a little bit enlarged, but just not enlarged. In that case, my personal approach is to go with interferon. Uh, whether that is Pegasus or Besremi, uh, either one is fine. And I generally do practice that way. If the spleen is enlarged, I am a believer of starting ruxolitinib early. And so I do tend to favor ruxolitinib when the spleen is enlarged, uh, to whatever degree. If it's not enlarged at all, like you described, and feeling fine, good blood counts, I think I lean towards interferon. Obviously, the HOPE PMF study would apply to such a patient. Um,
Mhm.
now the question about TP53. Well, that's never a good actor, right? Unfortunately, it's never a good news. But in MF, as you pointed out, oftentimes the VAF is low, and these clones may not do anything for a long time. But regardless, it's a little disconcerting. Yes. Um, and we don't know what exactly to give to hit that clone. We do not know that. We know that interferons are particularly good for the JAK2 clone. We just don't know what the right answer is for the TP53 part of it. Okay, but again, keeping transplant sort of ready, if you will, like, at least having a donor identified and having that process started, watching the clone closely are the things I would say. But we don't have any evidence-based drug that we know will help with a TP53 mutation, really.
Okay. And then this is probably going to be our last question just due to time. I know that you've got a hard stop in a couple of moments, but this individual said that they've heard there's a second type 2 JAK inhibitor in trials. Do you happen to know the name of the study or what that drug may be?
Actually, no. I'm aware of multiple companies, multiple, more than two, being interested in mutant selective, okay, in mutant selective JAK2 inhibitor, but I've not heard of another company besides Ajax with a type 2 JAK2 inhibitor. You may be right, but I'm just not aware.
Okay, that is good to know. And then I think that's going to wrap up our questions for today. So thank you so much, Dr. Bose. It was a pleasure to work with you and have you on. Um, so many exciting trials that you highlighted. So really, truly, thank you for your time.
You're welcome. Thank you all for listening.
Absolutely. All right. And then let me get back to my screen here. I did want to let everyone know, and I'll try to drop a link here in a moment. But HealthTree has recently launched a clinical trial finder that will help through the use of AI to help you figure out what trials you may be eligible for. So if you're interested in a clinical trial, I do recommend checking that out. If you have your medical records connected through HealthTree, we are able to help identify trials that might be a good option for you. So I'll drop a link to that. And if anyone's interested in that, please feel free to reach out to me because it is a really powerful tool and a great option if you're interested in these trials. But that being said, I wanted to let everyone know about some of the upcoming events that we have. So this Friday, we have Finding Strength After Setbacks, Real Stories from Relapse and Refractory Blood Cancer Patients. This is part of our Blood Cancer Awareness Month series. So really cool opportunity to hear from a couple of other members of the community about that experience of being relapsed and refractory and just kind of what that looks like and some good advice. And then after that, we have understanding myelofibrosis care options if you're not a transplant candidate. So again, this will probably cover some clinical trials, but just some other options for those of you who may not be eligible for transplant. And that'll be happening on September 23rd. And then we're going to be doing a Gather community event. This is an event without a doctor speaking, but really just an opportunity for the community to come together and speak with one another. And this event is going to be focused on share your symptoms and how you combat them. So really coming together, talking about how everyone feels and maybe what you do to help improve those symptoms. So highly recommend checking those three events out. Um, and then if you're interested in keeping the conversation going today, I will be staying on for a couple of moments and happy to chat with any and all of you. Hopefully we can also get some of you chatting with one another. But I did also want to let you know about our Myelofibrosis Connect group. This is a social media platform that we run through HealthTree. So it's for patients and really just a great opportunity for you to connect, that's the name, with others who really understand what you're going through. So if that's of interest of you, highly recommend checking that out as well. The QR code on the screen can take you there. If not, I will include that in our follow-up email as well. But with that, I just want to thank you all again for coming. Big thank you to Dr. Bose for sharing with us today. It was wonderful to see all of you. Please fill out that survey. It really does help us when trying to plan more events. My email is on the slide here. Feel free to reach out to me with any and all questions. I'm always available to members of the community. Um, and if you're gonna stay on, give me just a moment. I'll turn the recording off and stop sharing screen so that we can just have a little bit more of a conversational setting here. But if you are gonna hop off, I hope you have a wonderful rest of your day.